This episode is sponsored by NordPass. Get 70% off a 2-year NordPass premium plan, plus you get an additional 1 month for free. Go to nordpass.com/scishow and use the code SciShow to get this offer with a 30-day money-back guarantee to keep your passwords safe and organized. Not all cancer cells are created equal. Some have mutations in their genes that make them better at getting around treatments like chemo or radiotherapy, which means a tumor can develop therapeutic resistance, where those treatments don't work as well anymore. And even just a few cells left over after a treatment can turn into another tumor. But new research might help scientists understand how cancer cells evolve their differences. And that could help them understand and combat therapeutic resistance or develop even more targeted therapies in the future. In this study, published in the journal Nature in December, researchers looked at acute myeloid leukemia, a type of blood cancer. They tracked cells in 3 slightly different mouse models of leukemia, where all the mice had been infected with genetically identical cancer cells. Researchers then used a technique called SPLINTER, that stands for single-cell profiling and lineage tracing, to track individual cells. That would let them see which specific cells were doing something different, and therefore could become the dominant cells that form new tumors. SPLINTER works by tagging the DNA of individual cells with sequences of other DNA called barcodes. Those barcodes let researchers see which bits of DNA are actually being used by the cancer cell. The barcodes are also inherited along with the cancer DNA, meaning the researchers can also track how the cell evolves by seeing which bits of DNA stick around. They found that even though all the cancer cells had the same genetics, they behaved differently in different mice. See, different cells can turn on different sets of genes, or use the same genes more or less compared to other cells, even if all those cells have the same DNA. And because turning different genes on means that the cell will make different proteins, cells that might look identical DNA-wise might actually behave quite differently. In this paper, the researchers were interested in understanding which genes were transcribed in leukemia stem cells. These are cells that are likely to become the dominant ones and end up forming a new tumor. Researchers saw that leukemia stem cells repressed genes that tell the cancer cell to decorate itself with molecules that the immune system recognizes. Those molecules match up with immune cells that keep cancer under control by killing potentially dangerous cells. Leukemia stem cells also expressed a gene for a particular tumor growth factor more. Basically, they were better at being cancer-y. This study is the first time researchers have been able to study how individual cancer cells with the same genes might behave differently. The hope now is that other scientists can use the SPLINTER tool to study other types of cancer, because this finding could be really important to the emerging field of personalized cancer treatments. There's a lot of interest in developing treatments tailored to the patient right now, but our ability to do so mostly relies on looking for genetic differences underlying their cancer or other disease. And what this study shows us is that researchers and doctors will have to consider not just a disease's genetic profile, but how those genes are used as well. In other body-related news, scientists might have just figured out the difference between hearing and actual listening. Like, are you actually actively listening to the teacher go on about the 7 Years' War, or are you just like, hearing it? Because there does appear to be a difference. In a study published this week in Cell Reports, researchers looked at how a variety of factors influenced sound processing in the brain, and that included things like how engaged you are with a task, as well as how much you move around, whether listening is actually rewarding in some way, and finally, your level of arousal, meaning how awake you are. See, each of those things have been shown by previous studies to actually change the activity of certain groups of auditory neurons, but it's been hard to understand how those factors work together across the whole brain. This study looked at the activity of brain cells in 4 different areas of mice brains while they took part in a listening task. Each brain area is thought to be responsible for processing sound in different ways, some more complex than others. The mice had been trained to lick a spout when a sound of a certain length and pitch played. In one phase of the experiment, mice would get a sugary drink as a reward when the sound played, but in another phase, they did not get that reward. The idea was that the mice would be less engaged with the listening task if they were not being rewarded for it. Indeed, the researchers saw a different pattern of brain activity depending on whether a mouse was engaged with the test. They broke this overall activity pattern down into 10 specific sub-patterns based on each of the different aspects of the task. That included engagement, but also how excited the mice were, whether they were getting a reward, and the actual movement from licking itself. And they saw that mice who were more engaged showed more activity in the area that deals with the earliest and least complex sound sound processing. This tells the researchers that being engaged with a task actually helps the brain process sounds better. But some of that brain activity came down to all that other stuff: reward, excitement, movement, and so on. Licking, for example, might have made areas of the brain release particular chemicals that gear up auditory neurons for listening. Basically, researchers say that based on all of the different kinds of patterns they found, actively listening to sound is a really complicated process. The next step is to discover more about how these different groups of neurons affect listening. And who knows, it might just help us figure out how to tune in better during class. Thanks for watching this episode of SciShow News, which was supported by NordPass. NordPass is a password manager that lets you securely store and access all of your passwords in one place. Maybe you're like me, and you have a lot of different YouTube channels that you need to log into. A password manager sure would help. And your passwords may not be as secure as they need to be. For example, NordPass's research, which looks at the top 200 most common passwords globally, suggests that tons of people use local sports teams for their passwords. NordPass can help you generate more complex and secure passwords and autofill them when needed, and you can use a master password to store all of them in a secure vault that no one but you can see. You can get 70% off a 2-year NordPass premium plan, plus an additional 1 month free if you go to nordpass.com/scishow and use the code SciShow to claim this special offer. And if it's not for you, they also offer a 30-day money-back guarantee.